Okay. Uh, all right, participants, a very good afternoon. I welcome you all to the session 3 of the day 3rd of the AICTE sponsored faculty development program. Our last eminent speaker for the day is Dr. Vishnu D. Rajput, Associate Professor from Southern Federal University, Rostov-on-Don, Russia. Dr. Vishnu earned degrees with good academic records in agriculture and biological sciences. He has completed his Ph.D. studies from Chinese Academy of Sciences, China, and postdoctoral studies from Southern Federal University, Russia. Presently, he is working as an associate professor, leading researcher at Southern Federal University. His ongoing research area is based on soil contaminations, that is, potentially toxic elements and metal nanoparticles, and investigating the bioaccumulation, geo-transformations, uptake, translocation, and toxic effects of metallic nanoparticles on plant physiology, morphology, anatomy, and the ultrastructure of cellular and subcellular organelles, cytomorphomatric modifications, and DNA damage. He comprehensively detailed the state of research in environmental science in regard to how nanoparticles, heavy metals interact with plants, soil, microbial community, and the larger environment. He has published a total of 180 scientific publications, 106 peer-reviewed, highly rated full-length articles. He has authored 8 books, 25 book chapters. All of these are Scopus indexed, 95 conference articles to his name. He is an internationally recognized reviewer, peer-reviewed 98 manuscripts for international journals of repute, and received an outstanding reviewing certificate by Elsevier and Springer Publications. He is an editorial board member of various high-impact journals. He received Certificate of Appreciation for year 2019, Certificate of Honor for year 2020 by Southern Federal University, Russia, for outstanding contributions in academics, creative research, and publication activities. He has also received the prestigious status Highly Qualified Specialist by Southern Federal University and the Russian Ministry of Internal Affairs. I welcome you, sir, once again to the program. Sir, now I would like to request you to take on the proceedings from here onwards.
Thank you, sir, for wonderful introduction. Good afternoon, everyone. And as sir told that today, this is the last session and I'm going to talk about nanotechnology applications and environmental risk, especially insight into toxic effect of metal-based nanoparticle on barley. So I'm going to start right now. Okay, so today we are going to discuss about actually 3 parts in this presentation. I try to combine like general overview of nanotechnology, environmental concern, and my own research finding in this area. So first of all, we will discuss about what is nanoparticle, type of nanoparticle, global market of nanoparticle or nanotechnology, number of products available in the market, companies, countries that involved in the agriculture related nanoproduct development. And after this very huge application in various fields, where does the nanoparticle go, how it accumulate in agricultural or edible crops, and then what is environmental concern. And after that, we will discuss about my research methodologies and how these nanoparticles affect plant, especially for morphometric, cytometric, or physiological indexes, anatomical and molecular indexes. And then we go for conclusion and future of nanotechnology research. So literally, if we talk what is nanotechnology, we can say that engineering of molecules. If we reduce the size of any particle up to 1 to 100 nanometer, we can consider it as a nanoparticle. So at this scale, any material which we reduce the size up to 1 to 100, it enable unique properties and novel applications. So why we are making very small particle? Because at this scale, the particle have new type of properties and several innovative applications. Literally, we can say the size of human hair is 60, 100 to 100,000 nanometers. So we can imagine how small these particles are. Types of nanoparticle, natural nanomaterials, because this is not very new that nanoparticle was not exist. It was before, because there are many biogeochemical or mechanical processes that already making nanoparticles. So we can call natural nanoparticle, nanomaterial. Incidental nanomaterial, a nanomaterial unintentionally produced as a result of any form of direct or indirect human influence or activities. A lot of such type of activities now going on. By that, unintentionally the small particles generate. Engineered nanoparticle, this type of nanoparticle we are human making for their own purpose, intentionally produced by human. And anthropogenic, I mean both intentionally or intentionally produced nanoparticles. Basically, we can categorize these nanoparticles in organic and inorganic. Organic quantum dots, fullerenes, carbon nanotips, and inorganic, for example, metallic and metal oxide. These metallic and metal oxide nanoparticles are widely using in agriculture and allied sectors. So basically, this also using as a nutrients and in fertilizers we will discuss further. So in agriculture, this nanotechnology is booming and making a really comfortable a lot of new approaches for sustainable development or sustainable agriculture. Not only crop production, but also it improving soil fertility and productivity, so nutrient use efficiency, different type of new fertilizers, nano enabled fertilizers and sensors, and also improving the seed germination and from all overall plant development. So we can say it's a boon for agriculture. And it's not only crop production, I mean diagnostic, a lot of new sensors developed. By that we can understand the requirement of plants nutrients or disease management, and also for new technology together with the plant breeding or and biotechnology. So a lot of applications in agriculture. And also these nanoparticle helping us to cope up the various type of biotic stresses in plant, such as drought stress. There are many nanoparticles which proved under drought stress with the combination of other soil amendment, and scientists found the good response under drought stress. Also in salinity stress, for example, application of silicon based nanoparticle enhance plant growth under saline condition to increase the chlorophyll proline amount in various plants. Also now we are trying to cope up metal stress, because there are many polluted or contaminated soil and it's very difficult to grow agricultural plant. So these nanoparticle either stop to translocate those metals available in the soil to plants, or it change the mobility or change or also change the location of accumulation. So these are the fields where nanotechnology or nanoparticles are widely using. Similarly, for nano remediation approaches for restoration of contaminated soil, this is also a new approach. By this, uh, we are using phyto remediation and bioremediation to nano approaches, especially nanomaterial with combination of hyperaccumulator plants or microbes. So in this case, we try to grow plants in polluted area with the application of nanoparticles, various type of metallic nanoparticle to improve the crop biomass of plants to absorb more and more metal from polluted land. Similarly with the microorganism also. So these were the applications, and now we try to understand this market of nanotechnology. So nano era began in late 1990s, and the global market for nano composite totaled 2 billion in 2017, and is estimated to reach 7.3 billion by 2022. I mean, it's a very fast growing field and growing at a compound annual rate at 29.5% for period of 2017 to 2022. So this is a rapidly growing area, and thousands of nano product including nano fertilizer pesticides are available in the market. So there are one website that having a record of these nano enabled products. In that database, it was listed more than 3,000 product in different category that contain nanoparticles or nano materials. So here are some graphs. There are number of products. For example, Germany have more contribution, more number of products, and also especially in the fertilizer. If we see the graph of fertilizer, number of products more. There are more number of products that have nanoparticles in case of fertilizers, nano fertilizers. Also, mostly now in agriculture, it going, for example, pesticide, herbicide, fungicide, in plant protection field. Now, there's a big question about environmental concern. Where this nanoparticle go and what will be if it the concentration increase in environment. So there are lots of application intentionally unintentionally in form of micronutrient and insect pest management, and it finally go to the soil. Till now we do not have real data about nanoparticle concentration in water and soil in larger scale. So there are some model based predicted modeling studies that indicate these two nanoparticle widely used, zinc oxide and copper oxide. The concentration is quite high in soil, uh, 3,194 microgram per kilogram, and also for copper in water. So this is just predicted concentration, but recently the application is increasing in agriculture. So crop by crop, year by year, the concentration might increase a lot because these nanoparticles cannot degrade. So this is also a literature-based study where we found how much concentration of copper-based or many other nanoparticle accumulated in edible crops. So this is a surprising, I mean, wonder that is very high concentration sometime accumulate these elements dissociate from nanoparticles, and this concentration, I mean, various crops which human eating, the concentration increasing more and more. So now the concern is if the application is increasing, it might have toxic effect on soil microorganism, or it may have more impact on plant also, because the first time when you apply nanoparticle in your agriculture area, maybe the concentration is nominal and you have very good result, positive results, but might after several application it may disturb your soil microorganism as well as your plant growth. So the information about biogeotransformation is very little, and how it could affect soil microorganism. It also very rare information, not very clear information. How about plant toxicity? Last 6 year I'm working on this area, and we have in-depth information how it disturb internal organelles of plants and many other cellular, subcellular mechanisms and physiological mechanisms. But about how, if particle go to the soil, how it will interact with another elements, the information is very little. So these are the concerning issues. So for that, we plan two type of experiment. For primitive and quick results, we did hydroponic study, and same time for realistic information, we conducted experiment on barley, on soil in the real environment. So in this case, we selected barley, and because barley is very widely growing in this area in Russia, and also this plant suggested as a model experiment, as an indicator, bioindicator for pollution. So in this case, if we go for morphometric changes, I mean visual changes because of high concentration of nanoparticle, is quite primitive in the both cases, in soil and in hydroponic. The comparison between clean plants, normal plants, and a stress plant is very clear. And the polluted plants grow in polluted condition is very in the size stunted, and root structure disturbed, and even root morphology. If in this slide you can see here a very good root hairs, but in this case it's disappear only. I mean fibrous roots disappear because of copper oxide nanoparticle. But for these experiment we use, we plant before using nanoparticle in soil and water, we purchase this commercial nanoparticle, and we also synthesize this nanoparticle in our center, and then we go for how it have hydrodynamic stability or dynamic light scattering in water and in solution then before applying. So these are the our nanoparticle characterization by various techniques. And also correct, this is a zinc nanoparticle and this is a copper nanoparticle in water in normal condition. So we mostly found the size below 100, nearby 50 and plus minus. Sometimes we see aggregates also. So these nanoparticles we applied in our experiment. Uh, the main preparation for we, because we mostly focused on uh, anatomical observation, ultrastructural changes. So we follow this method, and we also develop our own method for holophyte plants, how we can make better slides for observing subcellular organelles by transmission emission microscope or scanned microscope. If we in as I already showed in one slide, accumulation from various literatures in edible crops, but in our experiment we also found very high level of accumulation of these particles in root, and of course it was higher in root and then in the leaf and in grain also. So this the concentration is very concerning. And after that we also tried non-destructive method to understand the elements, understand the nanoparticles in leaf. So for this we prepare flatten the leaf and then go for x-ray fluorescence. We just scan the leaf, and we just pointed the patches where we found high accumulation of these particles. Especially this particle accumulate in the leaf in the elemental form, dissoluted from your nanoparticle. This process is still not well known how it accumulate in which form. Then we go for ultrastructure. In first slide you can see here very clear root hairs, but in stress plant with the nanoparticle this root hairs disappear. So it shows that there is damage. And similarly for aerial part, I mean stomata, in first slide we can see very clear stomata, well organized and open. And similarly in stress plant we have damaged stomata. So it all connected with the plant development, plant process, stomata connected with photosynthesis. And here also a trichome morphology also disturbed in stress condition. And also we observe chlorophyll fluorescence emission. This is the normal leaves, and this stress leaf with the nanoparticle, the plants we cut. So for this experiment, we put our plant in the dark place for 20 minutes, and then we observe video record. We recorded video and then go for analysis, and we found the emission and compare. So here, if we see it disturb chlorophyll fluorescence, by that we can understand the quick state of plant. I mean, very quickly, if you want to understand if that does plant is stressed or not, because sometime by visual, plants look similar. Maybe chlorophyll content also seems similar. The color also seems similar. But if they have stress or they have nanoparticle stress, is very quick method by that, and a non-destructive method for photosynthesis. Also, we observe for vascular bundles, I mean, anatomy of root. So here we found the number of vascular bundles decreased, but the size increased. If we see in this slide, the total number of vascular bundle is decreased, but size due to stress increased. Similarly for leaves also. And then we go for ultrastructure. The ultrastructure of leaf, here we can see in the control plant, well organized mitochondria, and but in stress plant, the christie of mitochondria, the structure of mitochondria also disturbs. And similarly we go for chloroplast. We in the next slide we will discuss. And here when we observe in depth within the cellular and subcellular space, we found the particles of nanoparticles. It's a dark electronic dense material, and by EDX we observe that this is the particles dissoluted from nanoparticles. So here we can see inside when we observe for ultrastructure. This is a chloroplast. So here in control plant, we see there are well organized thylakoids, but in pollution it disturbs. So, and also on the another E and F, normal starch granules, more than 2, more than 3. But in stress, in one cell, the number decreased, but the size increased because very less consume of starch during stress condition. And it also have disturbed cellular membranes, and also accumulation of nanoparticle damaged. And also it disturbed the plastoglobulins, because these all subcellular organelles associated with the photosynthesis process, or all physiological process connected with these subcellular organelles of cells. So now the overall conclusion of this work was that the metallic nanoparticle, which is widely using in agriculture, may have or disturb the all chlorophyll fluorescence emission, deformation of stomata and trichome morphology, alteration to the cellular organization, irregularities of chloroplast, disruption to grana and thyla organization, damage to the structural organization of photosynthetic apparatus. Also is disturb the translocation of minerals from the root to shoot, and deviation in anatomical indexes connected with the physiological observation. And we also did the cytomorphomatric analysis, because by visual maybe it's not very clear to understand. So we go for cytomorphomatric analysis, and cytomorphomatric analysis of these all organelles also indicate huge damage in these organelles under stress condition. So when we go for gene, for example, transferase gene, it was also in a recent study, it was also disturbed. So what is the importance of these work to observe the physiological, anatomical, and morphometrics parameters? Because by that we understand how we can modify our nanoparticle, how we can coat our nanoparticle, because it's not possible to stop application of nanoparticle, but it is necessary to know in depth how it could be toxic, on which organelles it could be toxic. So that organelles, maybe we design our particles in the way where it can have minimum damage to the plant. The study about thylakoid organization connected with chlorophyll emission or scanned spectroscopy. By that we may understand if we reduce or we coat our nanoparticle, may not reach in these organelles, may only reach to the required area where plant need elements. So there are still gaps that there is no reliable method to detect, identify, and quantify NPs in presence of natural, even in natural environments. So knowledge regarding the behavior and potential toxicological risk of nanoparticle in soil is emerging. Because as in some slide I said, if nanoparticle exist in soil, there are chances to interact with another elements or with microbes, and it may generate new potential toxic elements. So for this, it need a long-term study on bio-geo-transformation and long-term study, long data about how it behave after 2 years, how it behave after 10 years. So in my opinion, my suggestion, the quantitative estimation of nanoparticles in edible part of food crops must be measured before supplying these products to the consumer or applying in the agriculture. Because there are some products, already I said you in the beginning slide, that more than 3,000 products available in the market containing a nanoparticle, but there is no long-term data how it interact with the soil organelles, so biota or plant organelles. In plant also have, there are minerals, maybe it interact, become more toxic. So these are still gaps, but still the products are in the market and going to the farmer fields. Also, it will be a crucial to develop a unified methodology for testing NP toxicity in environment, because all one nanoparticle may enhance plant growth in particular crop, but the same concentration may have toxic to another plant. So there are requirement to have larger scale studies. So another suggestion, what future of the nanotechnology research? So it is required to have a green synthesized nanoparticles, especially metal waste, because metal waste nanoparticle is widely used in for agriculture. And maybe this bacteria could have possibility to synthesize nanoparticle which is eco-friendly, cost effective, less toxic, so and can apply in agriculture or environment, in food industry or medical. So thank you for your attention, and if you have any query or question, we may discuss because we have enough time.
Dear participants, if you have any questions for this presentation, you can open your mic and can ask questions directly to sir. Yeah, dear participants, if you have any questions regarding this presentation of Dr. Vishnu, one may open their mic and can ask questions directly to sir.
So okay, thank you very much. I think nobody have
Yeah,
any
no
query
one.
regarding this.
Yes, sir.
Sir, shall I ask the question?
Sure, ma'am. Sure, ma'am.
Shall I ask?
Yes, ma'am. Sure.
Yes, yes, please, please.
Sir, I would like to know ki, although I am not very, yes, sir. Thank you for giving me the opportunity, sir. Sir, I would like to know ki, you are talking about the nanoscience and nanotechnology. Although I am not familiar with with this field, but yes, in future I would like to explore it. I am basically working with the biosurfactants
Mhm, mhm.
generate the nanoparticles of that biosurfactants.
I do not very well understand the question, but of course, because for example, organic metal framework, I don't know exactly about your area, but the main question now in nanotechnology, the solubility of these particles and stability and coatings. If we have just like a medical that some nanoparticle express in particular area, this is the current requirement because a lot of changes happen in nanosynthesis, in green synthesis, but the main task is to smart delivery, especially for plants.
Okay, okay. Thank you, sir. But
Mhm.
again, I would like to ask कि biosurfactant is a basically a bacterial metabolite, which is produced by a few microbes. So, if you see the functional capacity of this biosurfactant, they are working in a domain of health industry, also pharmaceutical industries also. So I would like to know ki whether we can generate the nanoparticles of these biosurfactant as it is a bacterial metabolite.
I think it may be possible if these biosurfactant can be combined together with the nanoparticle and may have some framework, and then maybe it enhance the uh, more impact, because I have no experience about
Okay,
exact synthesis,
okay.
but maybe this is the possibility if these
I
particles, some
know
suitable
I'm familiar
Mhm.
with the
Sorry.
yes familiar with the production. So I'm familiar how to extract it from the bacteria.
Mm-hmm.
I'm familiar with the identification part. So as
Mm-hmm.
the size of the molecule is quite large, say about 150 or 100 kilo daltons. So I just want to know with the help of the nanoscience, can we reduce the size and it is
No,
going to
may,
show a promising results?
maybe if you reduce size, maybe property change, but in my opinion, if you combine these your molecules with some nanoparticles, with to increase the impact, it will be more better rather than reduce size. Of course you can, but maybe in that case you may have different property or different I mean, it is a matter of experiment or research,
Yeah, yeah. Okay,
because how it
okay.
will behave after reducing the size, because it's a molecule, a
Yeah,
lot
yeah.
of, I mean,
True,
things change.
true, true, true. Sir, is it okay if I ask your email ID so that in future if I'm
Yes, yes,
yeah
yes,
if
yes. Rajput.vishnu@gmail.com.
okay, okay.
Also if you
I
search
would
also if you search my name, because I have social profile, and
okay, fine.
no I try to motivate the students from India, and there are many, more than I think 5,000 students connected with me, and sometime I try to be online to help them how to write a research article, how to get admission in foreign
Okay,
countries, especially in Russia. So
okay,
maybe it is more better if you connect there also. Every year we have possibility for postdoc, no, four
okay,
five postdoc we are recruiting every year, and some people working distantly due to COVID, and some already here. So these are the possibilities also, and I am always happy to collaborate, because this is not
okay.
all my research which I presented here. We are also working on polyaromatic hydrocarbons, also working on heavy metals, also we are working on biochar, also we trying to combine nano and biochar together for soil amendment. Such
Okay.
type of fields also we are working.
Oh, thank you so
So
much,
may
sir. Maybe very soon I will write you a email, and
You're
I
welcome,
would
ma'am.
like to take your guidance so that I can work in a desired platform with my PhD as well as with my M.Sc students, sir.
Sure, sure,
I am
thank you.
Dr. Shikhar Raikwad from MIT WPU, Pune, Maharashtra, sir.
Oh, very nice to connect
Yeah.
with you, and hope to connect in future as well. Thank you
Yes,
so much.
definitely. I will write one mail to you, sir.
Sure,
Thank
sure.
you so much, sir.
You're welcome. Thanks.
Yeah.
So there is
sir,
a
may
question.
I ask one question, please?
Yeah, sure, sure.
Sir, is that possible that nanoparticle enter inside the plant through pollution
But
method? If
if,
possible, then what changes are possible?
sir, if you don't mind, can you please introduce yourself first and then ask the question?
Sir, is this possible that nanoparticle enter inside the plant through pollution method or natural method? Then, if possible, then what changes arise in the possible factors?
Is actually is depend on the first depend on the size of particle and property, and on which method, for example foliar applications and inside soil application or any other metal another method if any. Yes, yes, sorry. I mean, any type of methods, if any particle enter inside the plant, there are changes in elements as well, because inside plant also have elements, also have nutrients and elements. So it have interaction with cellular and subcellular organelles, interaction with cell sap. So it may be toxic and sometimes maybe not, because it's depend on the particle size. But if higher particle size may have difficulty to enter as well. So the changes I already told in plant, we have now in-depth information how it could affect internal anatomy of internal biochemical or physiological or molecular changes. But still there is gap. We don't know how exactly it translocate and how it change their form inside the plant.
Dear participants, any more questions from your side? Dr. Vishnu would be happy to answer them. Those participants who are unable to unmute themselves can ask questions on the chat box. I'll read the questions for them. I guess there are no more questions from the participants.
Hello, sir.
Yes,
Hello.
Yes,
ma'am.
yes, please.
Sir, I have one question. All these experiments which you did, wherein you had seen the absorption from the aqueous solution, isn't it?
In soil also, we did in soil also, because we
Soil
spiked
also.
soil.
So my
uh,
question is how the absorption of nanoparticles from soil varies with respect to absorption from aqueous solution, because there will be great differences in the environment and the heterogeneity that is seen
right.
in the soil environment. And also, I believe that the nanoparticles might be bound to the matrix and clay minerals present in the soil, and that is why the availability of nanoparticles to be taken up by the plant roots will be quite different as compared to that in aqueous
In
solution.
You are absolutely correct. In both cases have different factors, but hydroponic studies are not the real presentation of real environment. But these studies just for to observe primitive results. For example, if the concentration increasing and
Yeah, then
in
what
existing,
happens? We get the
yes,
idea.
what
Yeah.
happened like this. And in the soil, of course, it have many another factors, no, not only soil physiochemical properties, soil biochemical properties also affect the mobility of particles. So there are many factors, but still we are applying nanoparticle in soil in case of fertilizer, even in the in box of where the manufacturer claim it may have a problem with the solubility also.
Yeah. Sir, I was working with nanoparticles and effect of these particles, this same nanoparticles I had worked 6 to 7 years back, and the effect on bacterial species I had checked. But surprisingly, what I found that either some of the particles, they were not having any effect on the growth of organism or survival of organism. Rather, some of them sometimes activated the organisms and their growth responses was found to be increased. So kind of no this surprising results I obtained rather than finding some inhibitory effect. But then I happened to find some of the papers which reported adverse effect of increasing concentration of nanoparticles on microbial communities of wastewater treatment plants, because wastewater treatment plants have a mixed microbial community, and their
Right.
activity
Some may
like
be tolerant.
nitrification, denitrification, everything got adversely affected by presence of increasing concentrations of these nanoparticles in the wastewater. So one review article also, I after searching a lot of literature, I had written one review article also on that. But unfortunately, in the laboratory experiment, I could not show any foolproof system by which we can show that the nanoparticles, yes, they have really adverse effect on the bacteria. That we could not show. So,
Actually,
have you come
yes,
across
please.
any kind of such observation, or have you read about it?
No. It's depend on the size. The main question about the size. If the size decreasing of nanoparticle, of course, on low concentration you may have positive impact, because I'm working on one experiment, and we have in this experiment we have biochar and heavy metal tolerant bacteria. We
Okay.
isolated and we identified these bacteria, and we put these bacteria with the nanoparticle in soil, separately with the comparative study with control and clean soil. And first year the growth was not very different, because
Hmm.
we inoculated our microorganism that is heavy metal tolerant, may have suppressed the toxicity of nanoparticle. But in second year results were different, so we trying to observe 5 year, because we already inoculated these microbes how together with nanoparticle and how it will be.
Yeah, yeah.
Maybe some stage the toxicity increase or decrease after several
Hmm.
years. So
Yeah,
such type of
and
finding
community
experiment needed.
succession may also be affected, I think. Succession of the species also
Right,
in mixed environment, no, will be different, right?
right.
Yeah, thank you so much. Very
Thank you
nice
so
work,
much.
sir.
Thank you very much.
Participants, any more questions from your side? Yeah, if any of the participants have any of the question in mind, can open their mic to ask the question. Great, sir.
Sir, may you please share your PPT or email ID, sir?
Yes, Rajput, RAJPUT, Rajput.Vishnu@gmail.com.
लाइट जला लिया यार अंधेरे में
At
लिखने का आदत पड़ गया
gmail.com.
है ये तो
All right, sir. Thank you, sir.
So thank you. It was really very how to say honor for me to connecting with all of you, and I am expecting some of you will connect with me in future as well, so we may have some very nice collaboration to work together. So thank you. Thank you, sir.
Thank you, sir, for this highly informative, knowledgeful, and inspiring session. Nanoparticles or nanoproducts are one of the recent explored products with innumerable usage in electronics and telecommunication industry, electrical industry, defense, agricultural industry for development of fertilizers, and also in biomedical industry and many others. The scientific arena has worked a lot on developing the methodologies for synthesizing of these nanoparticles and nanoproducts and exploring their applications in last few of the decades. But the recent challenges of nanoparticles as a pollutant of the ecosystem has opened new doors of research and development for effective and efficient management of the nano waste from the environment. With this, I conclude the session for today. Thank you, sir, for your gracious presence
Thank
for
you.
FDP. And I
Thank you,
wish
sir. Thank you.
best wishes to you and all the participants of the FDP for today. And thank you all the participants for the patience listening. Tomorrow again we will resume the session with for the day 4 of this FDP sharp at 10 a.m.
Thank you very
All the participants to join the session from 9:45 a.m. onwards. Thank you, everyone. Dear participants, Dr. Vishnu has shared his Facebook profile in the chat box. All those participants who are interested to connect with Dr. Vishnu on the social media can just click on the Facebook link and can follow him for the more of the updates and for connecting with him. Thank you.